Hi, my name is Dawn. I'm an OpenSciEd teacher, writer, and facilitator. I'm here to introduce you to Unit 7.3, Metabolic Reactions. In the science storyline approach used in OpenSciEd, the sequence of activities is designed to make sense to students. These storylines were developed based on the input from hundreds of classrooms and the questions students actually asked. So let's dig in and see how the story of 7.3 unfolds. OpenSciEd units are organized in collections called lesson sets. A lesson set is just a group of lessons that is focused on figuring out a part of our phenomena. At the end of a lesson set, students put their ideas together to explain what they can at that point. In this unit, there are 3 lesson sets. The first lesson set takes us through Lesson 8. Students investigate what happens to the food we eat, and does that work differently in McKenna's body? Across this unit, we follow the case study of a middle school girl named McKenna. In Lesson Set 2, now that we have figured out in Lesson Set 1 how a healthy digestive system breaks down food into smaller molecules which are absorbed through our small intestine, and how this works differently in McKenna, we want to know more now about how problems in one body system can affect other body systems. And finally, in Lesson Set 3, now that we know how a human body uses chemical reactions to get energy from the food we eat, we wonder if this happens in other animals. Do their bodies use chemical reactions to get energy from the food they eat? Then we apply what we have figured out to explain why bears survive the winter when they don't eat. Let's begin by taking a closer look at Lesson Set 1. In Lesson 1, our anchor, we hear from a girl named McKenna about some symptoms she's been having, and we read her doctor's note. Using this information, we develop initial models and ideas about what's happening in McKenna's body and what body systems we think are being affected. We identify related phenomena, generate questions for our driving question board, and brainstorm ideas for investigation. We're left thinking McKenna's symptoms have to do with her digestive system, but we have a lot of questions and want to know if there's any way to see if there's anything different inside McKenna's body. So in Lesson 2, we examine endoscopy reports from McKenna and a healthy person and analyze food graphs showing what happens to food as it travels through the small intestine of McKenna and a healthy person. We figure out McKenna's small intestine looks different than the healthy person's. It's smoother looking, and when food moves through the healthy person, different substances in the food decrease as they travel through the small intestine. But in McKenna, some of the substances don't decrease as much as in a healthy person. So now we want to know more about the small intestine. How does it work? In Lesson 3, we carry out an investigation using dialysis tubing to represent the small intestine. We use this to investigate why food molecules seem to be disappearing in the small intestine, like we saw in the data from Lesson 2. From this investigation, we're able to argue the small intestine must have microscopic openings in the wall of it, because small food molecules are able to get out, but larger molecules are not. Next, we analyze images of glucose and starch molecules, which makes us wonder more about what is happening to these food molecules in other parts of the digestive system, like the large intestine. So in Lesson 4, we analyze food data of a healthy person compared to McKenna in different parts of the small intestine and then into the large intestine. We figure out that in a healthy person, food molecules disappear as they move through the different parts of the small intestine, except for fiber and water. We see these seem to move into the large intestine and eventually leave the body as poop. We think the food that seems to disappear is actually being absorbed in the body. In McKenna, we see her poop includes more than just fiber and water. It also includes glucose, starch, and fatty acids. So now we're wondering, what happens with other complex carbs in a healthy person? Are they changing somehow? We decide we need to look at other parts of the digestive system. So in Lesson 5, we start in the mouth by analyzing data about what happens to other complex carbs other than fiber. We analyze data from a graham cracker again and see complex carbs and glucose change in the mouth. In order to find out more about what is happening in the mouth, we taste a cracker, chewing it slowly, and notice a change in taste. It seems to get sweeter. So we read about what is in our spit or saliva, and we find there's a chemical called amylase. we carry out an investigation with different types of food and amylase to collect data about what is happening in our mouth. And we figure out a chemical reaction is happening, changing some complex carbs into glucose because we see the amounts of complex carbs go down and the amount of glucose goes up. We wonder if this might also be happening in other parts of our digestive system. So in lesson 6, we analyze food data through the whole digestive system, noting how food changes in different parts of a healthy digestive system. We found the same pattern we saw in lesson 5. When some molecules decreased, others increased by the same amount. We figure out these changes are due to a chemical reaction happening in different organs of our digestive system. We are now ready to see if we can explain what is happening in McKenna's body. So in lesson 7, we begin by creating a gotta have it checklist to capture everything we figured out so far in this unit. We then take part 1 and 2 of a 3-part summative assessment. In part 1, we read about 5 different diseases and their related symptoms. We are able to narrow down the disease McKenna might have, but still need more information about the structures of the small intestine to conclusively diagnose McKenna. So in lesson 8, we begin by analyzing some microscopic images of the lining of the small intestine and encounter the word villi as the name of the small finger-like structures along the wall of the small intestine. In order to figure out more about these villi and what they do, we use a simulation and find when villi are longer, more food is absorbed into the blood. This leads us to figure out that longer villi lead to more food absorption and shorter or flatter villi lead to less food absorption. With this new piece of data, we are able to diagnose McKenna as having celiac disease. We then take stock of the questions on our driving question board and realize we still have questions about symptoms in other parts of McKenna's body, and so we want to figure out more about that. We're ready for lesson set 2. In lesson 9, we look back at McKenna's symptoms and take note of which ones can be explained by what we have figured out so far and which ones can't. This makes us wonder if maybe she's not gaining weight because her small intestine doesn't absorb as many food molecules. So in lesson 10, we analyze trends in McKenna's weight along with images of weight loss over time. We notice in the images it looks like fat is going away or disappearing, but where's it going? We carry out an investigation where we literally burn fat, vegetable oil, and duck fat, and see that when fat is burned, mass goes down. Now we want to know, though, what is actually happening when fat burns. So in lesson 11, we carry out another fat burning investigation, now in an open and closed system. We figure out that when the fat is burned in an open system, the mass goes down, but when it's burned in a closed system, the mass doesn't change, but the substances in the system change. Fat decreases, oxygen decreases, energy increases, carbon dioxide and water increase. This makes us wonder if this is happening in our body to provide us energy. Does it actually burn fat and other food? So in lesson 12, we know we breathe in oxygen and we breathe out carbon dioxide. This is similar to the burning we saw in lesson 11, where there was fat and oxygen before burning and carbon dioxide and water after burning. So we observe multiple sources of data, such as oxygen and carbon dioxide in the blood at different locations of the respiratory system, and glucose in the blood over 24 hours, and figure out that chemical reactions are happening in our cells. And when these chemical reactions happen in our body, glucose and oxygen is rearranged into carbon dioxide and water and releases energy that the cells can use, the body can use. This is called cellular respiration. In lesson 13, we take stock of everything we figured out that is happening in the different body systems within a healthy body by developing a revised model. We compare our models and develop a consensus model to explain how a healthy body uses energy and how McKenna's body could be functioning differently. This leads us to wonder if the same process is happening in animals. And this is the end of lesson set 2. Now we're ready for lesson set 3. In lesson 14, we investigate an organism of our choice to see if metabolic reactions happen in its body, similar to the way these reactions happen in humans. We figure out all animals, including humans, rearrange matter and food through chemical reactions to release energy. Some animals have different structures inside their bodies to perform these same functions, and some other living things, such as anaerobic bacteria, don't need oxygen for chemical reactions to get energy. In Lesson 15, we begin by returning to our driving question board to see if we can answer all our questions now using what we figured out. And then we use everything we've learned to apply to a transfer task to explain how bears can survive the winter without eating. And this brings us to the end of our unit. Thank you.